Hi children, in this video I will be dealing with the topic polysaccharides in the chapter biomolecules. So what are biomolecules? Biomolecules are also called biological molecules. They are produced by cells and living organisms. Now the important characteristics of these biomolecules. Biomolecules are macromolecules. Macromolecules, in contrast to micromolecules, have larger size and high molecular weight. Second feature, these are polymers. Polymers are chains of repeated units called monomers. And these polymers can be of two types. They can be homopolymers or heteropolymers. Homopolymers comprises similar repetitive units or similar monomeric units, whereas heteropolymers consist of a chain of different monomeric units. Third feature is they are all organic compounds. And lastly, they are very important for the survival of the cell and hence living organisms because they perform important biochemical reactions that regulate the proper growth and development of the body. So these are the very important four features of biomolecules. They are macromolecules, they are polymers, they are organic compounds, and they are very important for the survival of organisms and cells. The four major types of biomolecules are 1. Polysaccharides, 2. Nucleic acid, 3. Proteins, and 4. Lipids. So these are the four major biomolecules. Polysaccharides are polymers of monomeric units called monosaccharides, and these are usually glucose C6H12O6. Nucleic acids are polymers of nucleotides. Proteins are polymers of amino acids, and lipids are polymers of the monomeric unit fatty acids or glycerol. So here I have focused at the monomeric units. So what I am highlighting in green are the monomers. So monosaccharides are the monomeric units of the polymer polysaccharides. Nucleotides are the monomeric units of the polymer nucleic acid. Amino acids are the monomers of the polymer proteins. Fatty acids or glycerol forms the monomer of lipids. So in this video, we will start off with polysaccharides. Polysaccharides, as told, it is a chain, linear chain of a number of monomeric units called monosaccharides, and the monosaccharide is glucose. This is the chemical formula and structure of glucose. You all are familiar, but I am zooming through the structure of glucose. Repetitive units of glucose form a polymer. See the structure of glucose? It has a pyrene ring with 6 carbons, and look at the hydroxyl groups 1, 2, 3, 4 and 5 hydroxyl groups, and look at the carbons 1, 2, 3, 4, 5 and 6 carbon. So the chemical formula is C6H12O6. So these form the monomeric units of the polymer called polysaccharides. So once again, what are polysaccharides? Polysaccharides are carbohydrates formed by chains of repeated units or monomers, which are usually glucose C6H12O6 linked by glycosidic linkage. So the type of bond is the glycosidic linkage. Now the definition for a generalized definition for polysaccharide is over. So what is a polysaccharide? It is a polymer of monosaccharides or polymer of glucose, and these monomeric units, glucose or monosaccharides, are connected or linked by bonds called glycosidic linkages. Now let's look into the different types of polysaccharides, 2 in plants and 2 in animals. Cellulose and starch is found in plants. Cellulose are mainly, we know it's a component of the cell wall, it's mainly found in cotton fibers, and starch, glucose C6H12O6 is stored in the form of starch in plants. And the next parameter deals with the monomeric unit. You all know the monomeric unit is simple glucose. So analyzing cellulose, it is found in cotton fibers, and the monomeric unit is glucose, and the glucose are linked by the glycosidic linkage to specify beta-1,4 glycosidic linkage. So that is all about cellulose. So once again, about cellulose, which is the most common polysaccharide, taking the 4 parameters. Where are they usually found? They are found in cotton fibers. Then the monomeric units are glucose, and the repeated glucose units are connected by the glycosidic linkage, to be specific, beta-1,4 glycosidic linkage. Next is starch. In plants, the glucose is stored in the form of starch, and here also the monomeric units are the glucose, and the glucose repeated units are connected by alpha minus 1,4 glycosidic linkage. So once again, talking about starch, taking into account all the parameters, starch is found in plants, that is, the glucose is stored in the form of starch. So we can say glucose stored form is called starch in plants, and the monomeric units is glucose, and the glucose monomers are interconnected by the glycosidic linkage alpha minus 1,4 glycosidic linkage. Next, moving on to the polysaccharides found in animals. We have 2 of them, glycogen and chitin. Glycogen, again like in plants, excess of the glucose is stored as starch. Here in man or in animals, the glucose is stored in the form of glycogen, and it's stored in the liver. So the excess glucose does not remain in the blood, it is stored in the form of glycogen in the liver. And the monomeric units are also the glucose, and the glucose, just like starch, is connected by the glycosidic linkage alpha minus 1,4 glycosidic linkage. So that is about glycogen. So once again, I am highlighting glycogen, taking into account the parameters. Glycogen is found in the liver. It is excess of glucose is stored in the form of glycogen in the liver. The monomeric units are glucose, and the different glucose molecules are connected by alpha minus 1,4 glycosidic linkage. Last, we have chitin. Chitin is found in the exoskeleton of arthropods. Then here the difference is the glucose found in chitin is not the normal glucose. It is a modified glucose or modified sugar, which is derived from amino. It's an amino sugar. So there is a difference. The glucose is N-acetyl aminoglucose, and the linkage is just like cellulose. these glucose molecules, aminoglucose molecules, are linked by beta-1,4 glycosidic linkage. So, once again, the fourth polysaccharide, which is found in the exoskeleton of arthropods, we know prawn, crab, lobsters have an exoskeleton made up of chitin, and this contains a special type of glucose monomers, which is a modified sugar. It's an amino sugar called N-acetyl aminoglucose. These glucose molecules are connected by beta-1,4 glycosidic linkages. So, we looked into the different polysaccharides, 2 in plants and 2 in animals. Of these 4 common polysaccharides, the most simplest polysaccharide is cellulose, and the most complex polysaccharide is chitin. Now, why do we say cellulose is the most simplest polysaccharide on earth? The reason is that, first of all, it is unbranched, just a linear chain of glucose, and not only that, it is a homopolymer. That is, the monomeric units are similar and repeated in a linear chain. So, the most simplest forms of polysaccharide is cellulose, and the most complicated, complex, hard, strong chemically and physically is chitin. The reason is, can go correctly opposite, it is highly branched, and the branches has nitrogen as side chains. Then it is a heteropolymer, that is, the monomeric units are different. Not only that, to add to this rigidity or the strength or making the chitin hard is the specialty of the glucose. So, I would like to pull this picture of glucose. This is the normal glucose, the pyrene ring as I indicated in the beginning, with 6 carbon, 1, 2, 3, 4, 5, 6 carbon and hydroxyl groups. Look, 1, 2, 3, 4 and 5. Okay, this is the structure of a normal glucose molecule, very simple. But here in this modified glucose, which is found in chitin, this aminoglucose in the second carbon compound, in the second carbon, see, this is 1, 2, 3, 4, 5 and 6. The second carbon, the hydroxyl, is substituted. I am removing this, I am substituting. This hydroxyl in the second carbon is substituted by an amyl group. What is an amyl group? It is acetyl amine group. So, once again, the difference is with respect to the glucose. The glucose found in the other polysaccharides are the simple glucose comprising the pyrene ring, a single pyrene ring with 5 hydroxyl groups. But here, one of the hydroxyl group is replaced by an amyl group, and because of this amyl group, the hydrogen bonds becomes stronger. At the bordering polymers. So, there are the side chains which has, it is a polymer which is branched. So, the hydrogen bonds which are connecting the side chains become very strong. Therefore, it is very hard to break down. Therefore, this chitin does not easily decompose. That is one reason, because of the glucose. And the added on reason, it is highly branched and it is a heteropolymer. Talking about the remaining 2 with respect to whether they are branched or unbranched, that is, starch and glycogen are also branched. Branched polysaccharides, this is highly branched, chitin is highly branched and cellulose is unbranched. So, with that I wind up polysaccharides. So, once again reiterating, what are polysaccharides? Polysaccharides are long chains of monosaccharides called glucose, which are connected by glycosidic linkages. In all you see glucose. In all the polysaccharides we see glucose, but in chitin you see a modified glucose. Then the type of linkage is glycosidic linkage. See, in all it's the glycosidic linkage, but only in cellulose and in chitin it's beta-1,4 glycosidic linkage, whereas in the ones which is storage form, it is alpha-1,4 glycosidic linkage. So the linkage is linked with the function. And lastly, what I told, the simplest of all polysaccharides is cellulose, and I gave you the reason. One, it is unbranched, linear. Second reason is it is a homopolymer, and the most complicated, complex, hard, stable polysaccharide is chitin. One reason it's branched, second reason is a heteropolymer, and third reason is the type of glucose that constitutes the monomer. The glucose should have 5 hydroxyl group, but one of the hydroxyl group is replaced or substituted by an amyl group, which therefore makes the hydrogen bond at the bordering polymers very, very strong. So, it's very difficult to decompose chitin. I hope you have understood polysaccharides. Thank you.